Dear students, Assalamu alaikum. In the last lecture, we were studying the renal clearances. The objectives of today's lecture are to learn about some of the other clearance methods, and towards the last part of the lecture about the dilution of urine. Estimation of glomerular filtration rate by inulin clearance. If a substance is freely filtered and is not reabsorbed or secreted by the renal tubules, then the rate at which that substance is excreted in the urine, US into V, is equal to the filtration rate of the substance by the kidneys, GFR into PS. Therefore, glomerular filtration rate into PS is equal to U into V. The glomerular filtration rate, therefore, can be calculated as the clearance of the substance as follows. Glomerular filtration rate is equal to US into V divided by PS is equal to CS, that is the clearance. Inulin is a substance that fits these criteria. Inulin is a polysaccharide molecule. It is not produced in the body. It is found in the roots of certain plants, and it is administered intravenously to measure the glomerular filtration rate. This figure shows the renal handling of inulin. In this example, the plasma concentration is 1 milligram per milliliter. Urine concentration is 125 milligram per milliliter, and urine flow rate is 1 milliliter per minute. Therefore, 125 milligram per milliliter of inulin passes into the urine. Inulin clearance is calculated as the urine excretion rate of inulin divided by the plasma concentration, which gives the value of 125 milliliters per minute. Inulin is not the only substance that can be used for determining the glomerular filtration rate. Other substances include radioactive iothalamate and creatinine. Estimation of glomerular filtration rate by creatinine clearance. Creatinine is a byproduct of muscle metabolism and is cleared from the body fluids almost entirely by glomerular filtration. Measurement of creatinine clearance does not require intravenous infusion. Therefore, this method is much more widely used. However, creatinine clearance is not a perfect marker of glomerular filtration rate because a small amount of it is secreted by the tubules also. It is calculated by glomerular filtration rate is equal to clearance of creatinine is equal to UCR into V divided by PCR. If glomerular filtration rate decreases by 50%, the kidneys will transiently filter and excrete only half as much creatinine, causing accumulation of creatinine in the body fluids and raising the plasma concentration. Plasma concentration of creatinine will continue to rise until the filtered load of creatinine P into GFR and creatinine excretion U into V return to normal, and a balance between creatinine production and creatinine excretion is re-established. If glomerular filtration rate falls to 1/4 normal, plasma creatinine would increase to about 4 times normal, and a decrease of glomerular filtration rate to 1/8 of the normal would raise plasma creatinine to 8 times normal. Therefore, the creatinine excretion rate equals the rate of creatinine production despite reductions in glomerular filtration rate. However, this normal rate of creatinine excretion occurs at the expense of elevated plasma creatinine concentration. The figure shows the approximate relationship between glomerular filtration rate and plasma creatinine concentration. You can see that by decreasing glomerular filtration rate by 50% will increase plasma creatinine to twice normal if creatinine production by the body remains constant. Next, we study the estimation of renal plasma flow by para-aminohippuric acid clearance. Remember, it is the estimation of renal plasma flow and not the glomerular filtration rate. Theoretically, if a substance is completely cleared from the plasma, the clearance rate of that substance is equal to the total renal plasma flow. The amount of the substance delivered to the kidneys in the blood, that is renal plasma flow into PS, would be equal to the amount excreted in the urine, US into V. Therefore, renal plasma flow could be calculated as equal to US into V divided by PS equal to clearance of that substance. But because the glomerular filtration rate is only about 20% of the total plasma flow, a substance that is completely cleared from the plasma must be excreted by tubular excretion as well as glomerular filtration. Now, there is no known substance that is completely cleared by the kidneys. One substance, that is para-aminohippuric acid, is about 90% cleared from the plasma. Therefore, the clearance of PAH can be used as an approximation of renal plasma flow. This figure shows measurement of renal plasma flow from the renal clearance of para-aminohippuric acid. PAH is freely filtered by the glomerular capillaries and is also secreted from the peritubular capillary blood into the tubular lumen. The amount of PAH in the plasma of the renal artery is about equal to the amount of PAH excreted in the urine. Therefore, the renal plasma flow can be calculated from the clearance of PAH. To be more accurate, one can correct for the percentage of PAH that is still in the blood when it leaves the kidneys. The percentage of PAH removed from the blood is known as the extraction ratio of PAH. It averages about 90% in normal kidneys. In diseased kidneys, this extraction ratio may be reduced because of inability of damaged tubules to secrete PAH into the tubular fluid. Assume that the plasma concentration of PAH is 0.01 milligram per milliliter. Urine concentration is 5.85 milligram per milliliter and urine flow rate is 1 milliliter per minute. Now, pH clearance can be calculated from the rate of urinary pH excretion divided by the plasma pH concentration. This calculates to be 585 milliliters per minute. If the extraction ratio for PAH is 90%, the actual renal plasma flow then can be calculated as equal to PAH clearance divided by PAH extraction ratio. That is, dividing 585 milliliter per minute by 0.5 gives a value of 650 milliliters per minute. The extraction ratio EPAH is calculated as the difference between the renal arterial PAH and renal venous PAH concentrations divided by the renal arterial PAH concentration. Total blood flow through the kidneys can be calculated from the total renal plasma flow and hematocrit. For example, if the hematocrit is 0.45 and the total renal plasma flow is 650 milliliters per minute, the total blood flow through both kidneys is equal to 650 divided by 1 minus 0.45 or 1182 milliliters per minute. Now let's study our second topic of today's lecture, which is the urine dilution. The osmolarity must be precisely regulated to prevent the cells from shrinking or swelling. We know this fact from our previous knowledge. Now, extracellular fluid osmolarity and sodium chloride concentration are regulated by the amount of extracellular water. And in turn, the total body water is controlled by fluid intake and renal water accretion. The kidneys can excrete urine with an osmolarity as low as 50 milliosmoles per liter or an osmolarity as high as 1200 to 1400 milliosmoles per liter. An equally important point to note is that the kidneys can excrete a dilute or a concentrated urine without major changes in the rates of excretion of solutes such as sodium and potassium. Now, this is a very important point, and I repeat it again that the kidneys can excrete either dilute or concentrated urine without major changes in the rates of excretion of solutes like sodium and potassium. And this is important because this is necessary for survival, especially when the fluid intake is limited in certain conditions. Now, as I mentioned in a previous slide, the total body water is controlled by two factors. Number one is the fluid intake, and number two by the renal water excretion. And kidneys can excrete excess water by forming dilute urine. And ADH, antidiuretic hormone, also controls urine concentration by altering the renal excretion of water independently. I repeat, independently of the rate of solute excretion. To understand this further, let's now study the changes in water and electrolyte balance when someone drinks 1 liter of water in 10 minutes. What happens is that the urine volume increases to about 6 times normal within 45 minutes. The total amount of solute excreted remains constant because the urine formed becomes dilute and urine osmolarity decreases from 600 to about 100 milliosmoles per liter. This figure shows water diuresis in a human after ingestion of 1 liter of water. Note that after water ingestion, urine volume increases and urine osmolarity decreases, causing excretion of a large volume of dilute urine. However, the total amount of solute excreted by the kidneys remain relatively constant. These responses of the kidneys prevent plasma osmolarity to decrease markedly if excess quantity of water is ingested. Therefore, after ingestion of excess water, the kidney rids the body of the excess water but does not excrete excess amounts of solutes. Now let's study renal mechanisms for excreting dilute urine. When the glomerular filtrate is initially formed, its osmolarity is about the same as that of plasma, that is 300 milliosmoles per liter. To excrete excess water, it is necessary to dilute the filtrate as it passes along the renal tubule. This dilution is achieved by reabsorbing more solutes than the water. But this occurs only in certain segments of the tubular system. Let's study the changes in the proximal tubule. As fluid flows through the proximal tubule, solutes and water are reabsorbed in equal proportions. So, little change in osmolarity occurs, and the proximal tubule fluid remains isoosmotic to the plasma, that is having the same osmolarity, which is 300 milliosmoles per liter. Changes in descending loop of Henle and inner medulla. Here, water is reabsorbed by osmosis. The tubular fluid reaches equilibrium with the surrounding interstitial fluid of the renal medulla, which is very hypotonic. Therefore, the tubular fluid becomes more concentrated as it flows into the inner medulla. Changes in ascending loop of Henle. Here, sodium, potassium, and chloride are reabsorbed. As this segment is impermeable to water, even in the presence of antidiuretic hormone, the tubular fluid becomes more dilute with the osmolarity of 100 milliosmoles per liter. Therefore, the fluid leaving the early distal tubular segment is hypoosmotic. Changes in distal and collecting tubules. Here there is additional reabsorption of sodium and chloride. In the absence of ADH, these segments being impermeable to water, the tubular fluid becomes even more dilute, decreasing its osmolarity to as low as 50 milliosmoles per liter. Therefore, the failure to reabsorb water and the continued reabsorption of solutes lead to the formation of a dilute urine. I end today's lecture here and will continue with the topic of concentrated urine in the next lecture.